This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1067, recorded on December 1st, 2023. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello. Oops, I stepped on my own line. Hello, everybody. The weather outside is snotty. There's no other way to describe it. The temperature is in the 40s. It's drizzling. It's depressing, but it's Friday, so Saturday will look better, I hope. But today we're getting snotty weather.
7 degrees Celsius and drizzling here in Manhattan. Yeah. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. Dixon, you immediately trigger a song to me. Oh, the weather outside is snotty.
Yes. Me too. I thought the same thing. Yep.
It's 69 here and beautiful sunny day. It's beautiful.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It's 50 Fahrenheit, 10 C. It's supposed to get snotty later, but right now it's just cloudy.
Right.
Who did I talk to yesterday?
So we're cloudy with a chance of snot.
That's right. Snot balls.
Alan, I talked to someone yesterday and asked them where they lived. And they said, we moved to Western Massachusetts. I know it's a big place, but I always associate that with
you.
I don't know where. Also joining us from, I don't remember where you're from, Texas A&M, Jolene Ramsey. What's
Is that
the
College
town?
Station?
That's
College
right.
Station.
College Station, Texas. Hello, everyone. Today, it's 66 degrees Fahrenheit, 19 C, and mostly cloudy outside. Pleasant weather for the 1st of December.
College Station, Texas. We should know because we were there for something or other.
For live TWiV recording.
Courtesy of Jolene.
Yep.
That's where
College
the
Station.
phage pillow came from.
That's right.
Yeah.
There's also a very large vertical farm just outside of town.
You know, I have to show you this. Hold on.
I've never had a chance to visit that vertical farm yet, Dixon.
Well, it's a privately owned establishment that makes vaccines using plants.
Mm-hmm.
I wanted to show you this, Jolene.
Holy cow.
What?
That's a big phage.
The
This
virus
is
that
this
ate
is a
people.
crocheted.
It's crocheted, and it was done by a student of Mark Martin, who was recently on his podcast.
Vince, you should turn that into a microphone. It looks like a microphone.
The thing is,
it's like a microbe. It's not a microbe. It's a microphone.
It's got a base plate, right?
Yeah.
Tail fiber.
Yeah.
And this is, you can stand it up. It's actually able to stand up.
I like it.
Icosahedral head. So, Jolene, which phage would this be? Could you tell?
T4.
It looks like T4.
It's T4, yeah.
Even got the colors right.
That's today's
works well to
favorite phage, it sounds like.
How do you know the colors are right, Rich?
Oh, it's obvious.
He's been scaled down to that size to take a look.
I see.
Let me just tell you. All right. So first of all, also there's Matters Microbial number 18, which just dropped yesterday. It's called Why Phages Rage with Stephanie Strathdy. Okay.
We're going through a phage these days.
We're going through a phage. The reason is today is all phage because Jolene is here. I
This
just wanted to tell
phage
you
never passes.
Phage never passes.
This is Phage Week, National Phage Week.
Phage passes all the time, but
her name is
getting more.
Ruth Eisenberg. She made this. Okay,
Ah.
I love it. It's really good, and it sits up on top. You can't see it, so but since we're going to talk about all phage today, I thought you might like to see that. And apparently, it's T4. Okay, a few announcements first. Let's see if you enjoy our work here on these programs, and I don't think it as work. It's a pleasure to do it, but if you enjoy it in some way, please consider supporting us. We depend on your donations to do all this work, to remove Dixon's breathing in each episode. Costs $50 per episode. I was told that last week.
Really?
Mm-hmm. Yeah, $50 to go through and take out your breathing. Between when you talk, you tend to go.
No. I thought I muted myself all the time now.
Nah, it's okay. It's okay. By the way, I have a cough button here. You can't. Oh, yeah, there it is. See, it's a cough button.
Oh,
As I'm talking and I push the button. All right, did I go away?
you went away.
did.
So let me try cough. Does it get rid of the cough?
Yes.
Yeah.
It's called a rapcorizone. That I don't understand. Cough blocks. BLOX. Anyway, Dixon, you cost $50 per episode. So if you'd like to help us remove Dixon's breathing, please go to microbe.tv/contribute. Doesn't sound good. We don't want you to stop breathing.
No, we don't want to.
All right.
We don't want to stop Dixon's breathing. We
I
just
think
want
I'll
it
just
to be
breathe
inaudible.
in from now on. How's that?
No, it's okay. I do want to get you a new microphone
You do.
one of these
Yes.
days that will
Probably help a lot.
because the boom next to your face. That's it. But that's
Lord knows where you're going to put it over here, though. I've got everything all
I
over
will
the
ask the
place.
Lord when I come. Also, I want to remind you the American Society for Virology is again providing Global Scholar Travel Awards to international virologists interested in attending ASV 2024, which is June 24-28 next year in Columbus, Ohio. Preference will be given to junior faculty presenting abstracts. This year, the application deadline has been moved earlier to November
yesterday.
30th. Oh, this is I don't need to say
I don't
this.
think you need to say this
Yeah.
anymore. The application deadline for the Global Scholar.
Wait. What's the abstracts are not due for this for the November, but will be due at the standard deadline. Okay, so there's a standard deadline of February 1st.
Right, for abstracts.
So we hope you put in your Global Scholar Travel Award yesterday.
Yes.
And if you did, you have to get your abstracts in.
And that's if you're coming from out of the country. The regular travel awards are happening on the regular schedule, which is on the ASV website, I assume.
It's February,
and the
right?
management regrets that Time's arrow only moves in one direction.
I'm not sure we really regret that.
I do.
I'm not sure I want to. I'm not sure I want to change that setting. Just like because of the other consequences.
Yeah, well, that's
might be some things I'd go back and redo, but
yeah. That's right. That's
right. Okay. I want to remind you that Amy Rosenfeld is seeking a research assistant. I don't know if she still wants me to read this. I heard she got a good applicant, but I'll read it anyway. Then I will ask her. This is in the laboratory at FDA, the Center for Biologics Evaluation and Review. Dr. Rosenfeld works on enteroviruses, including cross-reactive antibody responses, making animal models, work in cell culture. So this is all done at BSL-2. And if you want more information, go to the show notes. I'll have a PDF there describing it some more with Dr. Rosenfeld's email address. And apparently people listen to TWiV and they apply. That's good.
Great.
It's good that a lot of people are listening. And I want to remind everyone as we approach this holiday season and you're looking for gifts, you should buy Dixon's book, The New City. You can go to despommier.com, the ultimate vanity website. And you can order a copy at Amazon, Barnes & Noble, bookshop.org. And, just order it. Dixon is in dire need of money.
This has nothing to do with my royalty payments.
This is to keep Dixon breathing.
Yes. Yes. Quietly. The one thing I'm missing in all of this is reviews of the book. And I know a lot of people have already bought it because I know what the sales look like from Columbia University Press. I just encourage people to render their opinion and list it on Amazon because that's also very helpful. So thank you.
We have an email from Martin. Dear Twivers, happy birthday to longtime Twiver, Dr. Kathy Spindler. Another one of the team reaching age 70 today. Yay, today is Kathy's birthday. Happy birthday.
Happy birthday, Kathy.
Happy birthday,
Happy birthday.
Best wishes to Jarl. Martin is in Cali, Colombia, where it's 20 C with sunny intervals and light winds.
Is Jarl an alternate spelling of y'all?
I don't know. It kind of puzzled me, too. I don't know what it is. And Rich, you have something for us?
Yes. I'd like to mark the passing of a virologist of note, colleague and friend of mine, Gert Suter, who died a couple of weeks ago. I think he was only about in his mid-60s, so a little early. Gert, I would summarize Gert's career, among other things, by saying that much of what we know about MVA, modified vaccinia ankara, which was initially a German smallpox vaccine that has been used now as a very much still a smallpox vaccine. It's the attenuated most attenuated smallpox vaccine, and also a vaccine vector. Much of what we know about that virus came from Gert. He was on the faculty of veterinary medicine at the Ludwig Maximilians Universität in Munich. He did his PhD there and then did a postdoc at Bernie Moiss's lab, where he initiated, I believe, his work on MVA, and then returned to Germany, once again to the LMU. Let me see. Oh, he became a leader. I'm reading from this obituary from LMU here. Became a leader of a research group in the Institute of Virology at Helmholtz, Munich. He obtained a habilitation degree in virology in Munich. Then he took up the chair of virology at the Institute of Infection and Medicine and Zoonosis at LMU and headed up the virology division at the Paul Ehrlich Institute. So a very productive career and just a wonderful person. I mourn his passing. Kathy has posted, there is a memoriam page on the ASV website with several people that we have memorialized in the past, and she has posted this tribute to Gert on that site, and we'll link both the tribute and the ASV site in the show notes.
All right, we have a couple of pre-snippets for you. This is something new. It's not a snippet. We have a snippet, but a couple of quick things to tell you. First of all, a page at WHO, avian influenza A H5N1 outbreak in Cambodia. 2 cases, November 24 and 25, of 20 to 25-year-old women who, one in 20 to 25, and the other less than 5 years old, who got infected with H5N1, and one died, the other recovered. This is interesting because it's a lineage of H5N1 that has been circulating for a number of years, at least 10 years. There's a newer lineage that's been spreading around the globe and breaking all the rules about seasonality that we've talked about before. But this is an older lineage.
This is not it. This is one that was kicking around.
It's an older code, but it checks out. I mean, it's an older lineage, but it checks out. So the report is quite interesting because they give you a lot of detail and a little bit of historical context. And then at the end, you go to WHO advice. They have a risk assessment. Just so you know, between 2003 to November of this year, there have been 882 human cases of infection with H5N1, including 461 deaths. It's about, 50% or so?
50%-ish.
From 23 countries. All of them, including these 2, have been linked to close contact with either live or dead birds that are themselves infected. And they don't spread from human to human very well, if at all. And they think these 2 individuals had 2 separate infections. So they say, it doesn't change the risk assessment, but when there are cases, they should be checked out. All right.
So
yeah,
Vincent,
worth keeping an eye on.
I want to just add an anecdote here. My wife and I have done a lot of traveling in that area.
Yeah.
Not recently, but in the last 10 years. And on one of our trips, we were floating down a river in order to see this wonderful homage to Buddha inside of a cave. And on the way back, they stopped, and we got out of the boats and walked around. And then on one of the posts was a stenciled skull and crossbones with H5N1 on it.
Yeah. What's the significance of that?
Well, I think they've probably found dead birds there.
Okay.
I hope that wasn't because somebody died from that.
Not dead people. Okay.
Yeah.
Cambodia. All right. And then we have another case, additional cases of chronic wasting disease in Minnesota. It's an article in SIDRAP. They now have a ban on farmed deer movement. Speaking of deer, this morning there were a couple of deer walking around in our neighborhood with arrows in their legs.
Oh.
Isn't that ridiculous?
That's
What do you do about
somebody
something
missed.
like that?
That's absurd.
I
They
mean,
didn't miss. They just hit the
They
wrong
didn't
part.
miss completely.
Right. They didn't. Yeah.
I mean,
They missed
it's
I'm
just-
sure they were aiming for the vital organs. And
I don't think you're allowed to use bow and arrows. I'm not sure, but that's really bad. I mean,
really?
yeah, they
There
probably
is a
ran
bow
away.
hunting season.
There's a bow season.
Yeah.
There is, but not that close to civilization.
Yeah, this is just
I don't know New Jersey's situation, but I actually usually bow season. I think a lot of places in Massachusetts, at least, it's before gun season, but it may.
That's right.
I think you can continue
Yeah.
hunting with a bow through the gun season, like that's allowed the whole time, but it's different from state to state.
Actually, so in New Jersey, there's 5 months of deer season. You can you could use a bow throughout that.
Okay.
Except I think you're not supposed to be in a place where there are just homes everywhere,
Correct.
right? On both
Right.
sides of the street. Anyway, this is a...
But deer move.
Deer do move, yes. December 23rd. What is this? December 23rd. This is
from 2019.
This is the wrong article. I don't have the right article, but there's a more recent one. I'll take that out because that's not it. There are additional cases recently of CWD in Montana.
Oh, here, I've got one. Yeah, so November 28th, 2023 expands into a new area of Montana.
Can you put that link in the show notes,
Yeah.
please? So there, we're seeing more and more of those cases. And finally, we have a CDC dashboard for wastewater sampling, which the first thing I saw were these nice drawings of pipes, pipes with something containing bacteria and viruses. It's a nice little graphic. So they tell you you can select different years, and you could look at COVID here, at least in the front page. So let's see. We have, the COVID wastewater is just going up here. There are other sites that can do this as well. And then they have one for Mpox virus detection in wastewater in the past 4 weeks. Look at that. There are quite a few dots.
A lot of those dots are blanks. 207 sites with no detection. 5 sites with intermittent detection. No sites with consistent detection. 417 sites with no recent data.
So these must be sampling sites that are established.
Yeah, they're sampling sites. What strikes me about the COVID-19 data is the apparent seasonality.
Yes.
Because I've been wondering about this. There seems to, I don't know if it's an assumption or whatever, that this disease is seasonal, and I'm not really necessarily close enough to the data, but I didn't know if there were significant data to support that idea, but certainly this wastewater sampling looks seasonal.
Starting to look that way, yeah.
A couple of weeks ago, we did a modeling paper on that, remember? I don't know if you were on, but
I was not.
They modeled seasonality of SARS-CoV-2 based on common cold coronaviruses, which differ themselves, right? Say they don't all have the same kind of pattern in the winter months, but if you model it on those, then it's going to be a winter disease, yeah. Okay, those are our pre-snippets. Now, Jolene will regale us with phage.
Yes. Well, by popular demand, there was a listener email about this first paper that we're going to look at last week, actually.
Yep.
And I had my undergrads coming to the lab after a break saying, "My dad asked me about this paper," and I was like, "Really?" So this is a paper that has received a lot of attention and is commonly known as the vampire phage paper. It's something in the ISME journal from Ivan Erel's lab, and there are a couple of co-first authors on this paper. The first two authors, I know Elia Mascola, who's listed as the second author, and then I believe this is a Portuguese name, Tajidi de Carvalho, something like that. Tajidi is the person who does electron microscopy and is very good at it, apparently. And there are several other authors, Stephen Caruso, who I know as a great educator in the C-phages program, Julia Lopez Perez, Kathleen Weston Hafer, and Christopher Shaper. These are individuals from the University of Maryland, Baltimore County, as well as the Universitat Autónoma de Barcelona in Spain and Washington University in St. Louis. So this story I know came out of the Phage Hunters program, which is a national initiative for students, primarily entering undergraduate students, but also many high school students have participated in this program to do environmental sampling and come back to the lab with those environmental samples and hunt for novel phages. And once they have those, to isolate them, sequence them, and do some characterization.
And one of the cool things about this program is that they very, very frequently find entirely new phages.
That's right. Yeah. Lots of new phages. The program started a long time ago now, and Vincent actually interviewed Graham Hatfull back when it was called FIRE before they partnered with the HHMI. And the Erel lab, Ivan's lab, was one of the early adopters of this program. And so they've been doing this for a long time. And this recent report that they put out is about some really cool new observations that they have made from these novel phages that have been isolated. So as they were looking for these phage, they noticed something different about this. So most bacteriophages that you isolate from the environment, you do this by bringing this sample in, and then you plate the bacteria on a Petri plate, and you make what is a solid lawn, we call it, of growth that is evenly distributed. And then when you add these bacteriophages, they will produce, not all of them, but some of them will produce these clearings that are often circular in nature that we call plaques. So as they were looking for these plaques, they found some phage that they had a hard time separating. So when you have an environmental sample, there's many different phage present in that sample, and you're searching for phage that infect the particular bacterium that you're growing. Here, they found that they had multiple plaque phenotypes, and then they dug deeper into that and found that one of these phage seems to be attaching to the other in a particular orientation that makes it look like the vampire. And so we'll go more into some of these details, but this happened to come out right around Halloween. And since one phage is attaching to the other in a position that kind of looks like the neck between where the head and the tail are attached, we have several different articles that have been written about it, including one for The Conversation that Ivan himself wrote, and there was a Washington Post article as well that was mentioned last week.
I would like to ask a question as a non-phage person, but interested in the subject. The searches that you just described, is there a host range for these phages that would be missed if you only used one species of bacteria, like E. coli or something like that? So is there a way of compensating for that when they look, because otherwise they're going to miss a lot of them, right?
There is a host range. Many phage are what we call specialists, and then there are generalists. And so if you have a generalist, then you're more likely to find that in the lab with the particular bacterium that you're hunting with.
Exactly.
And that's because the bacteria are very diverse in the surface features that they display. And those surface features are the receptors that the phage use to enter the cell. So you do miss a lot of diversity in a particular sample. We've reused the same sample many times with different bacteria, and you'll pull out different bacteriophages.
Sure.
But once you have an isolate that you're working with in the lab, you can test that host range, looking at different related organisms to capture some of that.
Gotcha.
Yeah, good question.
Okay.
Okay, so to kind of broaden this a little bit, what they initially start talking about in this paper is something called satellite viruses or satellite mobile genetic elements. So it turns out that this idea that one virus or one nucleic acid really is being dependent on another for its spread in the environment is something that has been observed in the past. And the classic systems in bacteriophages are known as bacteriophage P4 and P2. And in this case, what you have is one mobile genetic element, in this case a phage, that is in the cell, and it's there, it is missing some of the components necessary to spread on its own. And when the second element comes in that infects that cell, then it produces those missing components, and the first one can take advantage of that, depress or repress the expression of the incoming one, and take advantage to get out and spread. So for P2 and P4, P4 is integrated either into the bacterial chromosome or sometimes as a plasmid in the cell. And when P2 infects, P4 encodes these proteins that redirect the capsid protein of P2 to form a smaller head. And that smaller head accommodates the P4 genome and not the P2 genome. It can also redirect the packaging machinery to use preferentially the P4 genomic DNA as well.
Parasites attract parasites.
Yes.
Yeah.
P2 can grow on its own. Is that correct?
That's right. Yes.
Okay.
Mm-hmm.
Oh.
Because I remember, my familiarity with P2 comes from back in the day when there was a coli geneticist in the neighboring laboratory, John Cronin, who was doing P2 transductions all the time. It was a way of getting genes from one bacterium to another because, as I recall, if you infect with P2, the coli host chromosome is degraded and you get P2 capsids that pick up pieces of chromosome. So you have a lysate that has packaged pieces of coli DNA in it, which if you then infect another coli, it's a way of doing a transduction.
Okay, I'm not as familiar with the P2 studies, but we use P1 for that all the time.
Oh, maybe I'm mixing it up. Maybe it's P1. I don't know.
That's okay.
Yeah, that's probably right.
Right, but that is a really important tool that was necessary in in the development of moving genes around in bacteria, and it's still used today.
It probably is P1. I'm probably misremembering it.
It raises a question as to what the selective advantage is to have a complicated system like this. I know there are helper viruses also, which allow some viruses to reproduce at a more rapid rate that are transmitted along with the larger of the two viruses.
Yeah, hepatitis Delta, for example.
Yeah.
Example.
The Sendai virus has a helper virus, no?
I don't know about Sendai, but then there's adeno-associated virus. AAV.
Right, so this must have some selective advantage then. Can you suggest what that might be?
I think the answer to that is going to depend on the type of dependency that the satellite uses the helper for. So we call the one that doesn't have everything that it needs to spread on its own the satellite, and the one that brings in the necessary component the helper. And in some cases, the satellite is missing the protein capsid to move to the next cell. In some cases, it's missing the ability to replicate its own DNA. In some cases, it just depends on what it's missing, what the advantages per se would be. And the fact that we have so many different variations of these satellite nucleic acids, which can be satellite phages, or they can be some other kinds that we call that are just chromosomal elements that we don't call viruses or phages, that I think that indicates that the advantage is going to be dependent on the niche that nucleic acid has found.
But do you assume that the P4 the one that's dependent upon P2,
Mm-hmm.
was at one time competent and then lost that competency and is now totally dependent on the other virus for its replication? I mean, how did this come about? I'm wondering what, how would you even start to think about the selection for this?
It's a good question. For P2 and P4, for P4 in particular, I think that has been studied. There are some good phylogenetic analyses of P4 as a plasmid versus being in the chromosome and looking at the differences between those over a wide array of samples. I don't know off the top of my head, but I would think that it's going to, again, depend on the system, and we probably see it going both ways. That sometimes it looks like gene loss and sometimes it looks like genes gained that allow them to, in this case, maybe P2 became independent, whereas before it was something that was...
Are there families of P4 phage? I mean, whole arrays of them that are dependent upon other viruses besides P2?
So there are definitely many P4-like viruses that we can identify in the sequence databases. And I don't know how many of them have actually been studied and how easy it is to pair the P4-like phage with its helper phage, just with bioinformatics.
So you could, some of those could be independent. They could be free living, so to speak, phage that don't need a helper virus, and others could be satellites, right?
Maybe, if they have the genes that are otherwise missing.
Right.
But Dixon, successful systems attract parasites.
Indeed.
As Eugene Koonin says, if something can cheat, it will cheat. And that's what these guys are doing. They're cheating.
I get it.
So in this case, I don't think I read the title of the paper. So the title is Simultaneous Entry as an Adaptation to Virulence in a Novel Satellite Helper System Infecting Streptomyces Species. So they were hunting with Streptomyces, and they had environmental samples from Poolesville, Maryland, and St. Louis, Missouri. So these were kind of separately collected, and they found two different phage satellite helper systems in Streptomyces mirabilis and Streptomyces lividans. And they first did the plaque isolation that I was telling you about, and they found, I think this is really interesting, one of the clues that you might have a satellite helper system is that you have those two different plaque morphologies, and you can isolate one of them, which would be the helper, but the other one you can't get pure because it's not able to replicate on its own. So in one of the supplementary figures, they show pictures of the plaques of the two different plaque morphologies on a plate, and you can definitely tell them apart. So they first sent off their phage DNA for sequencing, and then they noticed the second clue, which was that they had two different phage assemblies come out of that, and one of them had a higher amount of the total sequencing reads attributed to it, and it happened to be the smaller one. So the two systems that they got out, one of them is called Mind Flayer and Mini Flayer, where Mind Flayer is the helper and Mini Flayer is the satellite. And the second one is Mulch Mansion and Mulch Room. And Mulch Mansion is the helper and Mulch Room is the satellite. So they noticed this difference in the amount of reads, and that makes sense if you have a satellite that is in a cell and the helper comes in, and then the satellite detects that it has what it needs to be able to move. So it's going to decrease the yield of the helper because it's stealing the capsid or the terminase or whatever it's doing. In this case, we think it's mainly needing the tail for its ability to transmit. So they saw both of these the difference in read numbers, the inability to purify the plaques, and then they went to look at the genomes. They both annotated the sequence that they got, and they looked at the particles themselves physically. So if you look in the paper, in Figure 2, they have several representative transmission electron micrographs shown here. They have just one of Mulch Room. There's no Mulch Mansion here, but they have several of Mind Flayer and Mini Flayer.
Apparently, they knew this was going to go viral.
Yeah, they did a pretty good job of spreading it around, I think.
Yeah.
So Mind Flayer has a nice icosahedral capsid and sipho tail that's kind of long and flexible. And Mini Flayer has a morphology that we call a short tail. So it's a podophage, and Mind Flayer is a siphophage. Independently, they look very nice, but not terribly unusual. But what they did see was a high proportion of the images. They had Mini Flayer right next to Mind Flayer, and in some cases attached, or it looked like it was attached. And they quantified this, and they found that in some of their purified samples, it looked like it was about 50% of the time they would see Mini Flayer next to Mind Flayer. And then they did a cool technique that I had not seen done before. This may be very common for other people who study viruses or phages. They actually usually we take a purified sample and put that on the grid and stain it and take it for microscopy. In this case, they took a little bit of media and put it, or sample solution, and they put it directly onto a plaque on a plate. And so that would have a mixture of the phages, and you wouldn't have gone through the purification process that might pull them apart. And then they put that directly onto the grid, and they saw a higher proportion of the phage on their grids were actually attached to each other.
So I have a couple of questions or observations. So they talk about how they couldn't separate Mind Flayer and Mini Flayer. They don't make the same comment on the others, Mulch Room and Mulch Mansion.
Mm-hmm.
But I kind of assume that the same association, biological association holds. That is, that you wouldn't be able to purify one on its own. And that they have, I guess I was confused because they had a picture of Mulch Room,
Yes.
but not the Mulch Mansion. And that made me think, well, how, did they somehow have that by itself? But I guess they can pick it out from a mixture. But then the question becomes, how do you know which is which in a mixture? And I'm thinking the relative abundance would be a clue, plus they're different sizes.
That's right. Yeah. So the head size is a clue
Right.
to what size genome it's going to have, because if you have a smaller head, you can't fit a bigger genome.
Right.
Right. So Mini Flayer's genome is 17,000 bases, and Mind Flayer is 130,000. So that's a big difference there.
Huge difference.
Now, in terms of the how they knew about Mulch Mansion and Mulch Room being a system like this satellite helper, if we look at the comparison of the genome between Mulch Mansion and Mind Flayer, which is in Figure 1, Part A, you can see that they share a lot of similarity among their genes.
Mm-hmm.
And you see the same thing for Mini Flayer and Mulch Room.
Mm-hmm.
And so that's a big clue that they are probably using a similar strategy, although there are some important differences between Mulch Room and Mini Flayer that may give some clues about some differences between how they interact with their helper.
The other thing I noticed is that the plaque size for the Mulch Room, Mulch Mansion thing was 0.1 millimeters that they reported. And I'm thinking, man.
Those are small plaques.
Are small plaques.
Did you look at the supplemental data, though?
Yeah,
they're not too hard to see.
It doesn't look too bad in the supplemental data. Right?
Sometimes there are some tricks you can use to make the plaques a little bigger, just based on the plating conditions.
Mm-hmm.
Yeah. Yeah, so they did some more bioinformatic analysis to look at how widespread this might be and how related they might be to other known phages. And it does look like these helpers are not too related to anything. These satellites are not too related to anything else that they'd seen before. And they did one more, what I thought was really cool analysis to get at the dependence of the satellite on the helper. And that was they noticed that the helpers have tRNAs in the genome. And that's a really common thing for bacteriophages. They bring in tRNAs. And we have several lines of evidence for what they might be using those for. And primarily, we think it's so that they can maximize the use of everything in the cell. And sometimes the host doesn't have all the tRNAs needed to make the proteins that the phage uses to build itself. So they bring along tRNAs to help with that translation process. And so they looked at tRNA usage for the major capsid protein, which is the protein that is the most abundant in the final built phage. And they saw that the codon usage for the satellites was more similar to that of the helpers than to that of the host, which is indicative that they are using similar, that the satellites are taking advantage of the helpers' tRNAs being present to produce the major capsid protein. Right. Okay. So that's kind of the data that they have presented here. It leaves a lot of open questions. If I didn't describe it really well, we've got this mind flayer, which is a regular syphophage with a head and a flexible tail, and then mini flayer with the podomorphology, but it's looks like it's physically attached to the tail of the sypho right next to the head. They have a little bit of indication that maybe there's an interaction between the heads, but there definitely looks like there's an interaction between the tails. That leaves a lot of questions about what is the use of this interaction in that particular position? How does the satellite get its genome delivered to the host cell if it's that far away from the surface? But they're planning to do future studies looking at how this system works.
Yeah, you have to wonder. I mean, obviously, the cell doesn't take up the whole phage. The head's left outside.
Right.
Mm-hmm.
They said they saw no evidence of any interaction between the satellite capsid and the cell.
Yeah.
So the preponderance of evidence at this point indicates that somehow the genome is getting in through this interaction with the tail. So I'm imagining this thing somehow inserting its DNA into the tail of the helper, which is a little hard to picture, but
yeah,
I agree.
Did they look? They probably didn't do it here, but you could easily look to see when you infect with this complex if both DNAs get in the cell, right?
They did not show anything for that here, but you could look. Yeah, it's
also if you get both cap. Wait, so they both have capsid coding genes of their own, correct?
correct.
So you could, if both capsids are produced in an infection, then that means that the DNAs are getting in, right?
Yeah.
So they make this comment, Jolene, this is the first description of a virulent satellite associated with a virulent helper. Is that because all the previous systems are temperate?
The majority of the helpers that have been described, they're temperate, meaning that the satellite system is associated with the host, either integrated into the chromosome or on an extra chromosomal element like a plasmid, and is maintained there until the helper infects. Yeah, P4
so temperate meaning it doesn't just kill the cell.
Right.
It sits there in the cell waiting for its ride.
And
right.
The reason this works is because they're attached to each other. Otherwise, it would be hard for a virulent system to work, right?
Yes.
Got
it. That's right. And if I remember correctly, one of them had mulch room contains a lysogeny module, so it is likely a temperate satellite, which would suggest that you wouldn't need this physical attachment between them.
Right.
Yeah, and there's this. There's these hypothetical genes in mini flayer that they were at least implying, or I inferred that those could be genes that I guess they had some similarity to structural proteins, that they could be responsible for this attachment to the tail.
Yes, I would like to see them do some mass spec on their purified virions.
Mm-hmm.
It's very cool.
Cool.
All right. Thank you, Jolene.
I love the pictures.
Yeah.
The pictures are really cool.
There were several that were modified, floating around on social media.
So, the term vampire virus, while I was thinking about this, obviously not knowing anything about it, I looked at that more like a remora on a shark,
Yes.
or a lamprey eel, which might latch onto something temporarily and then let go. You could do experiments here. This sounds like you could do some really elegant experiments by putting the competent virus on first and then shearing it off at various times after you've added the helper virus, and see whether or not there's a timing for this, as to whether both genomes have to go in together or they have to go in sequentially, or that sort of thing. Just like they did in the very beginning to see how a phage works, right? I mean, some of those early experiments back in the 60s and 70s were quite elegant for their time. And if this is going on in nature, though, it's really, there's a stoichiometry to this that just boggles your mind. How can anyone succeed if this is your modus operandus? And it obviously does. So it's there must be some affinity for that little zone where the other virus attaches to too, right?
Yeah, well, Dixon, that's why. Go ahead.
I would hypothesize that the satellite requires the tail of the helper to be present to really assemble a productive particle.
Okay.
I wouldn't imagine that you could attach afterwards. I think you'd probably have to do it at the same time.
What if you could get the tail of the competent virus only? And attach that to the bacteria?
I was going to say that. So you can, frequently when we purify phage in the lab, you get bands, typically in a gradient, and some of the bands have fully formed competent phages, and then others can be just heads that fell off the tails, and others can be just tails alone. So you could easily get tails.
So, and would that attach by itself to a bacterial surface?
It probably could, yes.
Do we know?
I have not looked at any studies where they use.
So if you
tails.
could take the tail of the competent, you're trying to find out. I'm sorry. Go ahead.
Go ahead.
Nope. I was just, back in the lab. How would you explore the requirements for the transfer of the helper virus genome into the bacteria?
Right. So you want to take the tail of Flair and see if you can get mini Flair to attach to just the tail and get its genome in.
Bingo. Mm-hmm. And you can do that experiment.
Yeah, they should be able to do that.
That's cool.
It turns out that there are these things called tailicins, which are basically phage tails that have no heads or other things that are used in bacterial warfare against each other. They can be secreted, or they can be,
So maybe the title of our show should be "Heads You Win, Tails You Win."
That's pretty good.
So these, Jolene, these were both originally soil samples. Is that right?
Yes, I believe so.
And Streptomyces, the host that they're using, is that a soil bacterium?
It
certainly is.
Okay.
Yeah.
That's where
so in terms of the natural environment, this is all going on in my garden.
Exactly.
Mm-hmm.
Yep.
Okay.
And yet it's so quiet outside.
You have to get little teeny microphones.
Vincent, you know that ivermectin came from a streptomycin species.
Yes.
Streptomyces avermidalis.
That made Rutgers University famous.
The avermectins. Oh, yeah. Yep.
I think people at Rutgers would probably argue that they had some claims to fame before that, but yeah.
They did. Well, they did. They did.
Selman Waxman, right?
Selman Waxman, that's right.
Dixon and I, we have a picture outside of Waxman Hall, I think.
Yeah. I knew his son. Byron.
Let's move on to eating phages.
Okay.
Yeah, I could do for some phage.
So this is a paper that I picked based on some work from a group in Australia, and this is something that is of interest to anyone who has who is alive, and that is because our microbiomes are an important part of our life, and our microbiomes are in all parts of our body. And in particular, we are thinking right now about the microbiome of the gut. There are thousands and thousands of phages in the gut. And those phages, the question is, how do we as human cells, how do our cells interact with those phages? We know that the phage interact with the hosts that are present in the gut, but do they interact at all with our cells? And this is a question that has been a big part of the focus of the lab of Jeremy Barr, who is the PI on the paper that I chose for this section. So this paper is called Mammalian Cells Internalize Bacteriophages and Use Them as a Resource to Enhance Cellular Growth and Survival. It came out in PLOS Biology just over a month ago. There are quite a few authors on this paper. It's the primary work of one of the graduate students from that lab who has now moved into a position of her own, Marion Bichet, who is now at Actalia and the University of Lorraine in France. And the rest of the team is at the School of Biological Sciences at Monash University in Clayton, Australia. There are also some individuals from Quebec.
RMIT University in Bundoora, Australia.
Right.
Mm-hmm. And Université du Québec, TWiV. Sorry if I butchered that.
No, thank you.
In Quebec.
And there are actually several departments from Monash University as well as the Westmead Institute for Medical Research in Westmead, Australia. So this group has been investigating the interactions of bacteriophages with mammalian cells for a little while, and they've had several previous papers. So one of them that is of interest is, first of all, this observation that they made that bacteriophages can be internalized into mammalian cells through a process called macropinocytosis. And the bacteriophages that are internalized are not all degraded. They are trafficked throughout the endomembrane system of the mammalian cell through the Golgi, and they can move. So a lot of our cells are present in layers that are polar. And in the gut in particular, we have an apical side of the cells, which are facing the exterior or the gut region where the microbiome is. And then there's the basolateral side, which is facing the rest of our body. They've observed that when they grow these cells, different kinds of cells in devices called transwells, where you can have separate compartments and polar cells in a layer in between. If you add phages to one side of that transwell, then you get transcytosis of phages across that layer. And it happens quite a bit, enough that they can estimate the amount of transcytosis that they get to be, I should have put the numbers here, in the many millions of billions of transcytosed phages in a given day for human cells. And they've also been able to see that those phages can get inside and they are able to infect bacteria as well. So they maintain their integrity. They're not just degraded, and they don't either completely activate the immune system. So this paper is kind of a follow-up on that, and one of the questions is what is happening to those phages that are getting inside of the cell? If there are between 10 to the 5th to 10 to the 6th bacteriophages in a mill of cells, what are they doing inside that cell? Are they affecting the cell in any way? So
I have some context, physiological context question.
Great.
Do you have any idea what the concentration of phage is in our guts?
There are estimates of the concentration of phage in our guts, and give me just a second. I put a recent review from the lab that put out this paper in the show notes, and I will include that link.
This is a mammalian cells guide on how to process a bacteriophage.
Correct. By Leo Kahn and Jeremy Barr. And they have some of these numbers in their review. So if I can find the right place, we know.
My problem here is that I'm a multiplicity of infection guy.
Mm.
And as I look at this paper, and they talk about numbers of phages and numbers of cells and stuff like that, I'm wondering, what is the
What's the
multiplicity of infection? Okay.
Well,
And I have it's not obvious. Okay. It's high, but it's
Right,
not
but
obvious.
it's-
And then
Well,
I'm wondering,
It's also not really an infection.
it's also not, there's not a single type
Right.
of bacteria or a single phage. So how do you calculate MOI for a mixed population of
Right, right.
bacteria?
And all these papers, by the way, are open access.
Right. Yeah, so we know measurements for human microbiome samples that have been measured through the fecal material is 10 to the 8th to 10 to the 10th phage particles per gram.
Goodness.
And that is a population that is continuously being overturned, but there is some stability to it. And the estimate that they made for how many phage get transcytosed in a particular or internalized in a body are 3 times 10 to the 10th in the gut.
Wow.
that's for the whole gut.
Yes.
That's 3 times 10 to the 10th per some unit of time.
Phages from the gut microbiome every day.
Okay.
Every day.
And those are known to circulate into many different tissues in our body. It turns out that you can find phage in almost any of the human tissue where you look.
Tissue or cells of the tissue?
Well, most of the studies haven't been precise enough to look inside the cells.
Okay.
Yeah.
So it could be the interstitial tissue, interstitial fluid.
It could be, yes. Mm-hmm. That's right.
So they're purposeful. That is to say, they're serving some physiological function, perhaps.
That's the question.
That
That's
is
the
the
question.
question.
Yeah.
That's the
Yes.
question right
Well,
there.
what happens
Are
if
they
you take them
doing
away?
anything?
Well,
What about
so-
a germ-free animal that has
Yeah.
no bacteria, therefore
Yeah.
no phage?
are they fine, Dixon? Or would you say they're fine?
No.
I didn't say anything. I meant to say that you can
No, I
actually
wanna know,
look by
because-
looking at animals without bacteria.
Because they exist, but maybe if you could pinpoint some phenotype that you could complement by adding back phage, right?
Germ-free
Yes.
animals tend to be very sick in a lot of different ways, is my
No,
understanding.
Yeah.
No.
Like
no.
I
they're
know
not
Dix-
they're not
the-
fine.
The reason I asked Dixon is because he used to work with them,
Yes.
and then he
Yeah.
shanks it. He says, no. He doesn't want to.
No. They looked perfectly normal, except that they all had very large ceca. The area of the small
Yeah,
intestine that
yeah.
joins up to the large intestine. For us, you'd have an appendix at that point. But the animals had very large. They looked like they were overweight, but they weren't. If you opened them up, of course, when you sacrificed them, you found all of this undigested fibrous material
Well,
that failed to transfer between the small intestine.
Yeah, that's not
Yeah.
a healthy animal.
let
That's
me say that
they behaved normally. They
the
picked you if they
the
wanted
editor
to.
of this paper is Ken Cadwell. Okay, Ken Cadwell published a paper years ago showing that germ-free mice, their intestines are screwed up. They have developmentally aberrant intestine, and there's no T cells in the intestine. And when you add back norovirus, it partially restores the developmental defects.
Ah.
So, maybe if you added back phage, you would partially, and maybe
So-
that's why he handled this paper.
When I added Trichinella to those animals, they behaved remarkably independent. Every animal gave a totally different number in terms of the infection, including 0 to over 200,000 muscle larvae if you waited a month after you infected them. So their gut tracts
Yeah.
are not uniformly screwed up. They are screwed up. I agree with you, but they're not.
Each is unhappy in its own way.
Exactly right. That's the way to put it. That's the way to
All
put
right,
it.
sorry.
And they're
Let's let Jolene tell us about the paper.
Yeah,
please,
Let's get back to
please.
Jolene telling us
no,
about
this
paper.
is very interesting stuff.
So,
Very interesting stuff.
Right. One of the reasons this is interesting is just because we are interacting with phage on a daily basis. And another reason is because we are thinking as a field about using bacteriophages as alternatives to antibiotics or as additions to antibiotics to treat bacterial infections in the clinic. And one thing that we know is that addition of phage, they have a very short half-life in the body. So once you treat someone with phage, they do not circulate for very long. So if they're being internalized, what is the cell doing with them? We know that this is primarily occurring via macropinocytosis, which is an actin-based process where cells will direct the actin to make kind of what's called a ruffle, and then they will form or engulf a large volume of extracellular material, and that includes these phage, and then those can be those macropinosomes can be trafficked and fuse with lysosomes, and the things inside can be degraded, or maybe some of that can be directed into a different trafficking pathway. And so one idea would be that the phage that come in get degraded in these lysosomal compartments and activate the immune system. So to study this,
the reason that would happen is because the cell doesn't like to have DNA in the cytoplasm.
Exactly. So toll-like receptors kicking and all that other stuff, right?
Exactly.
Okay, can I ask a spurious but maybe relevant question? Do mycobacteria have phages?
Yes, they do. In fact, the majority of the C-phages program was against hunting for mycobacteriophage.
Interesting, very, because they've got a mycolic acid coating, right?
That's
and these
right.
things go right through it.
Mm-hmm. They are adapted specifically for the surface features of their host.
I love it. This is great.
Yeah. So this group they used T4 primarily as their indicator phage. So where's that T4, Vincent?
Yeah, so this is the indicator phage. That's why the head of it is red. You can follow it.
Have I got T4 in my guts?
If you don't have T4, the sequence that's in NCBI, you have lots of T4-like phages.
Okay.
I promise you that.
Thank you.
So they use this as an indicator. It's a relatively large myovirus, very easy to grow. They have a nice protocol that Jeremy developed while he was a postdoc in Forest Rowers Lab called Phage on Tap to be able to purify large concentrations of phage and remove some of the contaminants from bacterial debris that is produced when you're producing the phage.
I looked that up, and I was really disappointed to see that it leaves out cesium chloride gradients. Because one of my fondest memories is cesium chloride gradients.
That was probably the biggest motivation for developing phage on tap.
Kind of the point.
Yeah, they're a pain in the butt, and they took big losses at that step. If I looked at the protocol right.
Yeah. Right. So,
but man, there's nothing like a band on a cesium chloride gradient.
They are beautiful.
Come on, their sucrose gradients were pretty good too, though.
I don't know.
I liked them. I like sucrose.
You also get nice bands on a sucrose gradient. So they had these purified T4 phages, and they had treated them carefully to remove extracellular DNA and RNA, and to remove the bacterial contaminants endotoxin. And we already know that can trigger the innate immune system. They also had some important controls. So phage are small, but they're not small enough that you can't filter them out. So they had purified phage, and then they took their purified phage sample and filtered it such that they removed the phage from that sample, and they had 4 times extra filtered sample. And that would be their phage-free sample to see that none of the other components in their sample were activating any innate immune system receptors. And then they had an additional control where they kind of broke open the phage and had components, protein, capsid protein, and so forth present, but not in its native conformation. So that they called their capsid control and their filter control. And then they had purified phage DNA, which had previously been shown in different systems to induce a response. So in this paper, they didn't use plaque assays for monitoring T4. They did use PCR. And the first thing that they did was they picked a couple of different cell lines. They're using A549 human lung epithelial cells and MDCK1 dog kidney epithelial cell lines. And both of these are epithelial cell lines because these had been shown in one of their previous studies to have a relatively high rate of uptake of phage by macropinocytosis. And they put these they treated these cells with their phage and controls, and then they did two things. They first watched them by microscopy, and they could see over a period of a couple of hours that they would get T4 phage DNA inside of the cells. And then they used several reporter cells that had luciferase reporters to detect the turning on of the C-gas sting pathway, as well as the TLR9 pathway activating interferon beta.
Both of those are sensing DNA, right?
Correct.
Yeah.
Yeah. So in both of those, in A549 as well as MDCK1, they did not have a significant amount of activation due to phage, either phage itself or filtered control. And then they took bone marrow-derived macrophages from mouse, and they looked at the production of murine P10 as an indicator for the detection of T4 in their samples. And they again saw no significant activation of these pathways in these cells. So even though they had treated these cells for 18 hours with 10 to the 7th PFU per mil of T4, they weren't really getting any indication that there was the triggering of an inflammatory immune response based on T4 DNA being present. Then the next thing that they did is...
So wait a minute. I have some questions about this. So what does that mean in terms of the fate of the phage that's getting in the cell? Does it mean that it just stays intact so the DNA is never exposed? Does it mean that it never gets out of a vesicle? Can you activate C-gas from a micropenisome? And I don't know that their controls necessarily address those questions because, a control would be to introduce DNA, but that's not necessarily going to go in by the same pathway, right?
Yes. So the control that they used was, they had different controls. For the case where they added DNA, they uses lipofectamine to add it. So it is definitely not going to be the same.
Right,
pathway.
but you've got the lysed phage. That would have exposed DNA, but how is that taken up? Do you know?
It presumably would also be taken up by macropinocytosis.
Pinocytosis, yeah.
And that...
That didn't induce, right?
That didn't induce.
It did not induce. Well, they didn't have the DNA. They don't show the DNA-only control here.
No,
they
that's
show
right.
capsid-only, filter, and T4 whole phage.
Right.
So another control that they probably didn't do either is they didn't denature the phage.
That's their capsid-only control.
Capsid only. I forget. Did they treat that to get rid of the DNA so that it's really just capsid protein? Because they heat treated the phage, right?
It's DNA treated.
Okay.
What about...
So that really is capsid only or capsid protein only.
Yeah, but what about heat denaturation?
That's how they that's how they ruptured the phage.
Yeah, right.
Heat.
Oh,
they heat denatured it, but then they DNA treated.
Exactly.
Right. So how can you maintain an intact phage particle anatomically and still render it neutral in terms of infectivity? How do you do that?
You can treat them sometimes with salt, high salt, or pH, and that will cause it to eject its genome, and then you get what we call ghosts.
No. There has to be... I'm asking a different question. The control for whether or not alive, and I know it's not alive, but at least it's infectious.
Active.
It's infectious. Phage particle versus one which cannot no longer cause an infection in a bacteria. Are those two different, or are they seen the same when you do these experiments?
You could irradiate it.
Yeah.
You could irradiate it.
Mm-hmm.
That's a good one. That's a very good one. There you go.
But that doesn't get to Rich's question, I think, which is what's happening? How are these things being pass through the
The
cell.
question is, these have DNA in them. Why
Yeah.
isn't that turning on?
Why
these
isn't that
pathways?
turning this on?
Is it because the phage is still intact, or is actually the TLR9 is in the vesicle, right?
Yeah, it is.
Yeah,
it is. It's inside.
Okay, so that suggests that the phage particles are intact.
Don't they do infectivity studies? And once it's in the cell, Jolene, they show that it's still infectious. Right?
Not in this study, but they did in previous papers. Yeah. So they know that they can recover a lot of PFU from the cells, meaning it's still active for bacterial infection. But not all of it is still active. And really, at the end of this paper, that's still a major question, is what is happening to these phage particles? We don't really know.
In the discussion, they say it suggests that they are tightly trafficked to prevent triggering the... okay.
There's also...
do they have a pass that says don't go to jail, collect $200? Yes. All the excuse me, all the other ones get caught. So what's the difference?
I think the idea is maybe they just stay in the pinocytosis vesicles and hang around in the cell for a while and then get let out.
There's no fusion of lysosomes, you say?
There is some fusion of lysosomes.
There is some, yeah.
And they did talk about that in their review. So they know that the pH itself, just inhibitors of pH itself, don't seem to have any impact. But if they inhibit some of the proteasome, some of the proteases, then they get less. They get more recovery. So it does seem like the proteases are doing some degradation of the phage.
So there's some
proteins
lowering of pH after they fuse.
When the macropinosomes fuse with lysosomal vacuoles or vesicles, then the pH will go down.
But it's not enough to neutralize all of the viral particles.
It is not. It's probably because not all of the macropinosomes are fused with lysosomal vesicles at any given time. Some of it's going to get trafficked through the rest of the endomembrane system before that happens.
And some of it's going to get out.
And some into the cytoplasm.
Right. So some of the phage that was pinocytosed in may then go back out.
Oh, yes, that's right.
Yeah.
Yeah. Usually in a polar cell, it would go back out the other side. But actually, that percentage is pretty small.
So it's just a passing phage.
That one is a passing phage.
But these phages, if they're entering the gut tract, they should be low pH resistant to begin with, right?
Right.
If they come through the stomach, yes.
Yeah, but if they're a lysogen, that wouldn't matter, right?
Yeah. Or if they're protected for some reason.
Yeah.
As they passage through. Yeah.
Yeah, a lot of I presume that most of the phage in my gut never went anywhere near the stomach. They're just resident in the gut and living there, turning over.
Yeah.
Right?
Mm-hmm. Right. But they had to get there somehow.
Initially, yeah.
Yes. Yeah. So in the next question, that the way they went about tackling this question of what are the phage doing inside the cell was they used two protein microarrays. So these protein microarrays were something that they purchased from a company called Connexus Biotech. And what Connexus does is they print these antibodies to many different proteins on a glass slide, and then they look for the presence of those proteins. And many of those proteins are not just the protein itself, but a phosphorylated version of the protein. And phosphorylation is, as we know, an important modification that indicates activity or inactivity of the protein in various different signaling pathways. So they had two of these protein microarrays, one that had 1,325 pan and phosphospecific antibodies, and another one that had 2,000 of those. So the second one was a little bit bigger. And they used those protein microarrays to analyze samples from both cell types. And they then did a deconvolution of those microarray data sets, which allowed them to map the networks and pathways that seemed to be activated or not in these cells after they had taken up phage compared to when they had not. This analysis, I'm sure, was a very big undertaking to try to make sense of, to put together what was really on and what was not. They had at least 52 hits for MDCK1 cells and 150 hits for the A549. And they focused in the rest of the paper on two particular pathways. Those were common between both the cell types, and that was AKT and CDK1. So in figure 2, they have network analysis diagrams for these proteins to show that CDK1 and AKT in both MDCK1 cells as well as A549 were changed compared to no phage addition. In the MDCK1 cells, they see that CDK1 is down-regulated, and they see that AKT1 pathways are up-regulated.
And CDK1 is like the cell cycle regulator,
Exactly.
Right? Mm-hmm.
So CDK is a cyclin-dependent kinase, and CDK1 is one of the most important central hub regulatory proteins for allowing cells to progress through the G2 phase of the cell cycle. So the cell cycle has if you start from cytokinesis, or you have two cells that have now divided into individual cells, and you start with this one cell, it usually goes through a growth phase, the G1 phase, and that can kind of arrest growth and stay in what we call a G0 phase, or it can progress from the G1 phase through checkpoints to make sure it's replicated enough of the materials of the cell to go into the DNA synthesis, or S phase, followed by the G2 phase, which is another growth phase, and then mitosis and cytokinesis again to divide. So CDK1 is important for progressing into G2. And what they took from this analysis overall is that this downregulation of CDK1 was holding these cells in the G1, G0 phase. So they were prolonging the initial growth phase of the cells by having taken up all of these phage. And they did not see that change at an early time point, which was 8 hours post-treatment, but they did see this 24 hours post-treatment.
Does this sound at this point as this might result eventually in an anti-tumor treatment?
No.
I mean, you want to prevent cells from dividing. You don't care if they get to the G phase, but you don't want them to go beyond that.
Eventually, they do divide, Dixon.
And eventually they're going to have to, right? Or they'll die. Yeah. But if this stops them from dividing,
this doesn't stop them in a way that you'd want to for cancer cells.
Yeah, it's not enough.
This pauses them, as we're going to get to in a moment, and it makes them healthier. Is basically.
No, we don't want to do that to cancer cells.
Yeah, no, this is what's happening here is actually ends up being a good thing for the cells.
Right. Yeah, so it suggested that they're growing more. So they used flow cytometry to quantify the number of cells that were in the different phases of the cell cycle, and they saw that there was a little bit more in the G1, G0 phase for T4 treated cells 24 hours post treatment. And then they also used another assay that is an indicator of cellular metabolic activity, or cellular proliferation, some people use it for, the MTT assay, which is a colorimetric assay.
It's looking at mitochondria activity or something like that. Is that right?
Yeah, that's right.
So it's, general metabolic activity.
Mm-hmm. Right. So in this experiment, they used T4, but they also used a few other bacteriophages. They used NS09, which is an uncharacterized virulent podophage. And then they used T3, which is another podovirus similar to T7. And then they used lambda, a particular variety of lambda called lambda vir, which is a virulent version of lambda. And so for all of these treatments, they noticed that only with T4 treatment and NS09 did they see the significant increase in metabolic activity compared to untreated cells or their controls. And
I have to say, Jolene, these differences are not huge, right?
they are not very large.
Cell cycle.
When I first looked at figure 3, I was like, oh, so there's no difference. Oh,
Yeah.
wait, there's an asterisk there. There is a difference.
They only see what they call a significant difference after 3 days of treatment. They did not see that after 1 day or 2 days of treatment. And the differences are not enormous. And the T3 and lambda vir are not considered significant, but they're closer to NS09 than to the control. I agree. It's not huge. It's a long time. But if you think about the scale of the number of phage that our cells are capable of taking up on any given day, then there probably will be a lot of phage. And it, in that case, might have a measurable effect.
And also, a small effect in a living organism can add up to a big deal. You know, it's okay, so these cells are doing a little bit better, but they're doing a little bit better every day. So,
well,
that makes a big difference.
I think you need to have a experiment in an animal to say, does this mean anything, right?
Yeah, because these are cancer cells. That
the cells grow.
These are immortalized
right.
cell lines.
That's right.
Cells are going to grow no matter what, with or without phage, no problem, right? It would be interesting to know if you took away some serum, right? Because you got tons of serum in these cultures. And
could they survive?
Actually, MDCKs will grow in serum-free medium.
Yeah.
Aha.
But they're also supplemented with other nutrients, right?
Probably.
Yeah, but the fact that some phages did it and others didn't suggests a specificity for the result.
Well, what if you just put protein in cells, big proteins that would get taken up by macropinocytosis? Would you have the same effect, right? I don't know if this is specific
They'd
for
be
phage.
degraded, though, Vincent. They'd be degraded right away.
Well, okay, but they'd be food.
Mm-hmm. Exactly.
That's
I mean,
true.
would they turn on these pathways? I mean, a lot of pathways were turned on. These are just 2 that they picked, right?
Yeah.
So I just
Right.
don't know if it's you add stuff to the medium, and it always happens, right? Pick a big protein. What's a big protein assembly?
Titan or something like that.
Yes.
So what is the ultimate fate of these phages? Do they eventually get digested and recycled through their metabolic pathways of the cell?
Well, that's what we were talking about with Rich's question originally, was what happens to them, and the answer is we're not entirely sure.
Exactly.
We know they're not turning on the
Well, what happens
DNA
when
response, but
no, but in those cultures, what happens when you stop feeding them the phage, and then maybe a week later or 2 weeks later, what does that look like?
Probably fine.
Where
'cause
are they?
There's cells in culture.
Where did they go?
Where did
Do you
the
remember-
phage go? Or how did the cells fare compared to untreated
both.
cells?
The fate of both the cell, the host, and the parasite. In this case, not a parasite.
it's not a parasite. The
No,
food, whatever it is.
It's
Cellular
a symbiont.
resource.
It's a symbiont.
Hmm.
Yeah, 2 different genomes working together to give a positive effect.
I don't think the genomes are working together. I think the phage is kind of an inert object
Alan,
in this.
I didn't mean that genomes are working together. You can't have a symbiosis with animals with the same genome. You need 2 separate genomes.
Yeah.
Then they can.
I don't think this is a
association.
symbiosis.
What do you think it is?
I think this is a meal.
Yeah.
Maybe
a long-term meal.
Well, do you remember the paper we did a while ago where they show that some protist can survive on just virus particles?
Yes.
Right?
Mm-hmm.
From Dave Dunnigan's lab. And so there, the cells are actually eating the particles. It's not clear that that's what's happening here.
They're
There's some kind-
phagophagic.
Yeah, but the thing is that the normal way of degrading material for metabolic benefit,
Lysosome.
this phage doesn't follow that pathway at all.
Well, these get into the lysosome, right, Jolene?
Yeah,
Yeah.
that's right.
Yeah,
It
they
didn't get
do.
degraded.
Not all of them, but some of them. And the important thing to realize, or I guess the argument that they're making in the discussion, is that phages are about 50% protein, 50% nucleic acid. And so that's a lot of nucleic acid that the cell doesn't have to put the effort in to make nucleotides. If they just pull out that phage DNA and use it as a resource. Maybe they're not...
It's a salvage pathway.
Mm-hmm.
Right?
Yeah, if they're being... It would be nice to know if they're...
Right, so the idea they put out in the discussion is maybe this is kind of a... I interpret it as kind of it's a reserve, it's this, they've got phage in the pantry so they can pull out some nucleic acids when they're ready to
I think we should add some labeled radioactively or whatever labeled, whatever people do anymore, and see if it gets incorporated into the cell.
Yeah,
right?
see where does it go in the cell? Not if, probably where. Where is it going? And how much? Is it funneling into a specific area? Yeah. Does it get into the chromosome in the S phase, or does it end up somewhere else?
If you irradiate the phage and make thymine dimers, among other things, then that will not be degraded as fast, and then the cells might grow slower as a result. You might want to try that. I don't know.
Well, the problem, Dixon, is that in serum the cells are growing like gangbusters already.
But of course.
Right, 24-hour cycle. So I don't know. Does this? You said it prolongs the cycle, Jolene, right?
Yeah. Well, it looks like it because more of the cells are in that cycle. Yeah.
Well, then you should substitute phage for the entire nutritional aspect of the medium.
And they live on phage alone.
See what happens. That's right.
I could live on phage alone.
You could. You do. You could make just medium, right? Add no serum to it, because that's what cell culture medium is. It's
then enrich with phage.
And you can actually do it without the amino acids, with nothing but just minerals, and then you add phage. You see how they do.
Right. Exactly.
Yeah.
Look at that. We could all apply for a grant right now.
No, I don't want to do
that. Yeah, I'm pretty sure this crew is doing most of the experiments we just discussed.
They should be. Yeah.
This is a long-term project and interest. And they're really, although there are many unanswered questions here, no one has done this systematically before. So this is the kind of study that we want to have if we're going to be adding extra phages of particular types to our body to know, what is the impact going to be, if any. And the first question was, is there a problem with the immune system? Not so much, maybe. If that's not the case, then are we going to be skewing anything in a way that we don't want? And maybe that's something we just prepare for as opposed to not knowing about. And then we do get a lot of variability in the kinds of experiments, the kinds of treatments that have been done, because we're using different kinds of phages all the time, and there are different bacterial pathogens, and humans have so much variability. So having a defined system to be able to study it in is a really big use.
Yeah, I mean, we're talking about all the experiments that could be done or should be done, and certainly from my perspective, I don't want to give the impression that I didn't like the paper. I think this is really important, great work. It's just at a very early stage.
I mean, it's all these phages are there. You should figure out if they're doing it.
Yeah, and one of their previous papers was looking at, for phage therapy, this has pharmacodynamic implications, because you're putting in a dose of phage, and you want to know where that goes. And if it just gets taken up by the cells and scrapped for, parts, then your therapeutic phage is not going to get very far.
So R2D2 walking out.
But I think
and this paper, I mean, especially that last experiment we just talked about, where they looked at a few different phages, it suggests, as Jolene just said, if you go in with different phages, you may see slightly different results.
Well, I think they ought to take an intestinal line, right, or Keiko cells, and use some biologically relevant phages.
Might be nice to see some primary cells, too. If you
primary cells. But I think the animal experiment...
T4 is a biologically relevant phage, I would argue. Yes. It's an E. coli phage. E. coli is a huge component of our gut.
But these are not gut cells that they're using here.
Okay.
Right.
Well, they're epithelial cells.
They are epithelial cells. Seeing this in a gut organoid might be interesting.
I want to see it in a mouse. I want to see if you give mice phage, will some aspect of their biology change? That's a big question because you don't know
So
what to look
salmonella is an intracellular bacterial infection.
Correct.
at. Mm-hmm.
This would be a good bacteria to aim these phage at.
Mm-hmm.
Can you cure an organoid gut of Salmonella typhi using T4?
That's a different question from this, though, right?
No, it's a totally different question. Of course it is. It's my question. No, I asked the question because when you start to think about the real world and what the infections are of the gut that eventually get passed on to the rest of the body,
Yeah. Yep.
then maybe these are another line of defense. And that's really important.
Yeah,
I mean,
classically,
really, really important.
we would think that intracellular bacteria were protected from phage, but that's not really necessarily the case.
Right.
Hopefully not. Otherwise, that would scrap a lot of people's grants, do you think?
All right, that was cool. Thank you, Jolene.
Yes,
Yeah,
thank you.
Very
wonderful.
cool.
I like that very much.
Wonderful.
All right, we need to do some picks now. We'll have to
Picks.
skip these
Okay.
letters. We'll do them next time. Dixon, what do you have for us?
Well, I've got my pick. I'm still focusing in on the environment, and the COP 28 is now in session in Dubai. And if you can imagine something as contrasted as a meeting whose sole purpose is to try to get the world to reduce their use of fossil fuels, and holding that meeting in the center of the production of fossil fuels, and then putting the people in charge of the production of fossil fuels in charge of this meeting. You read this article in
It
the
sends a
Times.
clear message, doesn't it?
It certainly does, and it raises so many levels of skepticism and cynicism that it's palpable. I mean, I don't blame this little girl in Sweden for going around and screaming her head off.
She's not a little girl anymore. She's
No, she's
a
not.
grown woman.
She's a teenager.
Still at it.
She's still at it, and she shouldn't stop. Obviously not. So when a country like China or Russia or the United States says we're bound and determined to cut our CO2 emissions through the use of reduced fossil fuel use, and then we start buying more fossil fuels and giving more grants for exploring for fossil fuels at the same time that you just said the other thing, then it's laughable, but it's also very sad. So this points out the true dilemma of climate change control. The true dilemma is that anybody whose ox will be gored by reducing fossil fuels, and those are the largest users, are not in favor of reducing it at all. They want to continue to use it, and they use it as long as they can until somebody forces them to stop. And who the hell is going to force China or Russia or the United
Or
States
us.
to stop? I said the United States. Who's going to force us? The citizens of these countries are not going to stop it.
Mother Nature is going to force us.
Mother Nature is already.
I do hope you're going to have some optimistic picks in this series, Dixon, because
Well-
there are actually some trends that suggests that it may not be up to these currently powerful people what happens
Let's hope not.
with the adoption of these technologies.
Send me the references in case I don't see them. And
There's a
like to-
tremendous uptake of renewables because
Oh yeah, no.
renewable electricity is cheaper now, and
Tell me
so
who
that
makes
just
Alan, who makes the most in terms of solar panels? What
China,
country? Yeah, exactly.
yeah, and they're installing a lot of them.
You know what? It turns out that they're talking out of both sides of their mouth at the same time.
Sure.
Yeah, but they have the
And,
biggest carbon footprint, right, Dixon?
That's right. Let's do this until we can't, and then we can do this. That's what it says.
I mean, by far, huge.
Yeah. Foxes guarding the hen house. That's the concept.
Foxes guarding the hen house. Very good. All right. Thank you, Dixon.
All
Rich,
right.
what do you have for us?
I have
Yeah, this is cool. I like this one a lot.
an electric vertical takeoff jet.
Right.
The company is called Lilium, and they are in the business of trying to develop and commercialize electric non-fossil fuel flight. And this is one of their prototypes. And it's what looks to be, I don't know, the plane itself can theoretically, at least, I don't know in actuality how it's configured in the prototype videos that are shown, but it can be configured to carry 4 or 6 passengers, and it's got electric turbine jets that are actually integrated into the wing flaps in a fashion that they can, as flaps do, change their angle relative to the wing. And there's a forward wing and a rear wing. There's no classical tail to this thing.
Yeah, this layout is called a canard wing design. One of the
wing?
major proponents of it was Bert Rutan, who built a series of aircraft on this kind of pattern. It's aerodynamically, it's very nice.
If you actually, they're not huge jets, but there's a lot of them.
Right.
If I counted them up, I count 3 dozen jets on this plane. 6 on each side on the forward wing and a dozen on each side on the back wing. And because the flaps can tilt seriously, this thing actually does vertical takeoff and landing. So goes straight up, flies some distance, and comes straight down. So I thought that this was fascinating. As I think about, the future of the planet, I wonder how we're going to, or if we're going to, be able to replace fossil fuels in air travel. And this is a step in that direction. I thought it was
I'm
really
so torn about these. I really... I mean, I grew up with flying car fiction and like, wow, that's really cool. But at the same time, NASA actually has a major segment of their budget called urban air mobility, where they're hyping this, and there are a dozen tech startups and companies who are trying to build similar or other concepts. And on the one hand, I say, wow, that's really cool. And on the other hand, I say, this is exactly what the world does not need. Because this is flying, especially vertical takeoff flying, is the most expensive, least accessible, most failure prone, most weather sensitive, most energy intensive way to get from A to B. And yeah.
Well,
it is awesome. And I think the technology in these is awesome. But at the same time, this is not how you reduce energy use. I don't care what your power source is. You power it on hydrogen or electricity. That still had to come from somewhere. And if you ran a train on that amount of electricity, you could carry 500 people, thousands of miles for the energy it's going to take to lift 2 people across town in this thing.
Yeah,
but anyway, it is very cool technology.
That's right. And as a tag to this, a recent other plane that can take off vertically and land vertically is the Osprey.
Yes,
and that
we just had one crash in Japan.
Another major accident. How many of these do you have to have?
Well, it's okay. The Osprey is a military. No, the Osprey is a military aircraft, so accident rates for that. I mean, they're doing stuff.
They're horrible, though. They're horrible. They're horrible.
They have a specific use. They should not be used for anything.
Nobody else but a Marine would ever get into one.
That's what they're for.
I know that. No,
Marines.
We see them flying up and down the Hudson River all the time.
Yeah.
But to know that they're that accident prone and they can't fix it because it's a design flaw.
Okay.
When the B-29 was first built, they blew up.
Oh yes, they had some bad
time.
Problems with
all
that.
the time. Yet they need to use them. Since we're talking about carbon, there's this cool pie chart, all the world's carbon emissions. So China 30.9%, US 13.5%.
Look at the difference.
And India is 7.5%. Those are the biggest ones. Russia's 4.7%. Yeah. Japan is.
Russia.
If you do it per capita, though, things skew a little differently.
I'm sure.
The U.S. doesn't look so great in that comparison.
Oh, you're right. But China, with all of its progress,
with 1 billion people in it, China is emitting a lot of CO2.
Yeah.
Yes,
they're still building coal power plants,
and they're still building coal power plants because that's what they have.
That's right.
Yeah.
So if you like sushi, don't eat it more than once a week.
That's why they made inactivated COVID vaccines. formalin inactivation. Alan, what do you have for us?
I have a book that I read a little while ago. Really fun read. It's called The Far Land by Brandon Presser. And this is, it's about Pitcairn Island, which you've heard of if you've heard of the HMS Bounty mutiny. This is where the mutineers went, and for about a decade, nobody knew where they were, because it was this island that had been spotted by one ship at one point, but had never been visited by anybody in Europe. The mutineers sailed around the South Pacific and found it and settled on it. And then sometime later, an American ship just happened by and discovered them, what was left of them. And there have been many, many stories told about this. What's really cool about this book is a couple of things. First of all, Presser put in the serious legwork and research to try to capture a perspective that's been missing from a lot of those stories, which is the Polynesian perspective. Because the mutineers on the Bounty, after they threw Captain Bligh and many of the other officers into a boat and sent them on the greatest small boat navigation accomplishment in history, they then went back to Tahiti, picked up the mostly women who they were interested in being with, and a few men to help with the crew. And then that's who went off to Pitcairn Island. So Presser actually talks about the culture of Tahiti and Polynesian, society generally at that time, and what the relations were like at that time. And also goes to the effort of finding as much as he can about what actually happened to these people and how they likely handled the situations. He reconstructs a lot. The other thing that's cool is you can go to Pitcairn Island, not easily. There's no airport. There's a cargo ship that goes there 4 times a year, and that's their contact with the outside world. That's their physical contact. They have, the electronic communication these days. So Presser went there, and unlike most people who, extreme tourists who actually go there, the thing to do would be to ride the ship there, visit for a couple of days until the ship is leaving, and then go back on the ship. Presser got dropped off, stayed for 3 months, living with the 2 main families on the island, who are descendants of the Bounty mutineers, and talks about that, the experience of actually being on the island when the ship goes away, and you're there at what some people have called a trailer park at the end of the world. And it's fascinating, So he alternates these perspectives: the historical, the timeline of the mutiny and what happened in the dynamics on the ship, and then the what he's experiencing going around the island and interacting with the descendants of it. It's a really cool book.
Nice.
There's just 48 people living there?
Yep.
Yes, I think the current population is 48.
Jeez.
Highly inbred.
Yes.
It depends how long they're at Dixon, right? My pick is a article in PNAS. It's open access. I was at Princeton yesterday talking about science communication, and I met a faculty member who really he's really into communicating science, and this is one of his favorite papers. This is a paper called Using Narratives and Storytelling to Communicate Science with Non-Expert Audiences. It's by Michael Dahlstrom from the School of Journalism at Iowa State. And so this, storytelling is a powerful way of conveying complicated narratives. He says here, he starts off by saying, this is very funny. Storytelling often has a bad reputation within science. Viewed as baseless or even manipulative, stories are often denigrated with statements such as, the plural of anecdote is not data. And Daniel says that all the time. Such a perspective is valuable within the context of scientific data collection to underscore the important difference between making informed generalizations from systematically sampled populations versus overgeneralizations. But when it communicating to non-expert audiences, the stories, anecdotes, and narratives are not only more appropriate, but potentially more important. And research suggests that narratives are easy to comprehend and audiences find them more engaging than traditional logical scientific communication. Once out of formal schooling, non-expert audiences get the majority of their scientific information from mass media, and they rely routinely on stories, anecdotes, and other narrative formats to cut, although the information clutter, and resonate with their audience. So this is an article all about, what is storytelling and how do you do it. And
So Vincent,
I just thought, one second, Dixon. I don't want to tell stories, but I think when we do a paper, we're telling a story.
that's
Although
what I was just going to tell
Yes.
you. I was just going to say you have a remarkable talent for that.
we all do, and I
No,
think
no,
that's
you do.
I think that we do tell stories, although they may be a little complicated compared to the narratives that he's talking about. But I think the most interesting paper discussions come when we say, well, here was the state of the field 50 years ago, and
right,
and this is what's, make a story out of it. I think it's fun. We want to talk about the people doing it and tell stories about them, right?
You bet.
So,
I would argue that stories are the way humans understand the world.
That's
This
true.
is not a scientist versus general public distinction.
You're right.
The stories we tell are different, but science is a story.
Here.
It is the whole thing. I mean, the deep technical aspects of the paper, that all fits into a story, whether you admit that you're writing one or not.
I agree.
And I think what Dahlstrom is saying in this paper, though, is he's talking about the kinds of stories that we would say, oh, that's just an anecdote. But he's pointing out that those more streamlined, simplified stories are what people outside of science are accustomed to.
yeah.
And what people who want to communicate science need to try to get to in order to communicate that science. And so there's a disjunction in the level of complexity of the story and the way it's structured. And I think he makes a very good point that you need to be aware of this if you're going to try and communicate science to a wider audience.
So I'm curious, does he talk at all about the importance or not of including a person in the story? Does there need to be,
I think either in this or in another paper, I think he goes into that.
Yeah. I think that's clear that David Quammen always says, if you want to tell about science, if you want to tell people about science, tell them about the people who did the science, and then people respond. I think
that's right.
I agree.
Every story needs a hero, and in science, the hero in the story is often the data.
Yeah.
But that doesn't resonate with people generally. I mean, even with us, I don't go to the movies to see data. I go to the movies to see, unless it's a Star Trek movie. I go to the movies to see, a story about some character I can relate to.
I think the stories of science are fascinating, right?
They are.
They truly are.
And any good science book is written as a story. I agree. And I just wanted to point out that I think we do that here on TWiV as well. Sorry, go ahead, Dixon.
No, my statement to you before was nobody tells a better story when they review a paper than you do.
No, don't say that, Dixon. We have a good crew here. They all do the same thing.
No, we all do the same, but you take the lead, and we follow your lead, because when you break it down into its components, you think like the people that have just conducted those experiments, and then they decided to, and then they tried this, and then they, it puts you in the lab with the people as the paper unfolds.
That's what I'm saying, is that I don't think we're so far off from storytelling.
Right. So yesterday I was at a school in New York City called Frank Sinatra School of Arts. And it was established by Tony Bennett with his fortune. And it's a public school, and people get into it by auditioning, either in the visual arts or in the performing arts.
Mm-hmm.
And the woman whose child goes there happens to be the publicity agent for Columbia University Press. So her daughter, was at the book signing, Vincent. You probably met her.
I remember her, yeah.
She was the one selling the books.
Yeah, I bought 2 books from her, yep.
So I went there yesterday, and speaking of telling stories, if you'll pardon me for just putting this in front of you, I challenged the students in all three classes that I addressed to try to put their finger through the table that they were sitting at. And I said, go ahead, try to, and they thought I was crazy. And I said, you all know why you can't do this, but it's probable that you can't tell me directly. You have 3 or 4 stories ahead of that, and then you can tell me exactly why you can't put your finger through the table. And I said, you know what a hydrogen atom is, and you know what the inside is. What part of that? That's the positive part. Oh, yeah. And then there, outside of that is the negative part. I said, that's an atom. I said, and those are all atoms. Are all atoms the same? Yes. Do they all have negative charges outside? Yes. They all agreed with me. They all knew that. I said, well, then so does your finger. And then I said, so does the table. And that's why you can't push your finger through the table. Now, that's story number 1, just to get their wheels turning. And I said, okay, where does the table come from? Now, they thought I was saying from trees. No. Where do the atoms for the table come from? And now remember, this is the Frank Sinatra School of the Arts. So I said, to find out, you'll have to listen to a recording by Joni Mitchell. So I got one of the students in the classroom to actually get the YouTube version of Joni Mitchell singing Woodstock. And in that song, you may not know this, or some of you may not know this, there is a reference to a Carl Sagan statement that says we are all stardust and we are billion-year-old carbon. Those are 2 lines from that song that she wrote for Crosby, Stills, and Nash to go and sing it at Woodstock because she couldn't go because she was scheduled to go on TV that week.
Yeah.
So you get all these intricate little stories tied into. I said, everybody knows the lyrics to that song. And if you just think a little bit about it, there's a lot of science in a very short sentence. And I said, that's what science really does. It takes a big body of information and condenses it into a sentence.
Yes.
And if we tell you that you're made out of star stuff, then you have to know how stars are formed, and then you have to know what happens to them afterwards, just like you have to know what happens to phage when it gets inside of a cell. What the hell does it do afterwards? What is it doing there, and why is it there? And if you could break away from your ordinary world and ask these esoteric questions, which are not so esoteric, then you can become a scientist. And I said, that's why I became a scientist.
You know what? One question that a non-scientist would ask after today's discussion that none of us would ask because we know the answer, maybe. Do the phage replicate in those cells?
Yeah, exactly. Exactly.
That was, I bet people would want to know. Yeah.
I got a quote for you.
Yep.
It comes from a poem by Muriel Rukeyser called The Speed of Darkness. I don't necessarily recommend the entire poem, but the relevant line is, find it here. I'm sorry. Well, it's the universe is made of stories, not of atoms.
That's right. Oh, that is exactly.
That's good.
That is exactly.
All right. We have a listener pick. Derek writes, TWiV, y'all might appreciate.
Did you skip? We have Jolene.
What?
Exactly.
You skipped over Jolene.
We have Jolene's pick, which I'm staring at on my monitor next to me.
Jolene, what do you have for us?
I have an illustration by David Goodsell. It's originally a watercolor painting called Bacteriophage T4 Infection. And this you can find in the Goodsell Gallery at the Protein Databank, which is a collection of structures of molecules that are publicly available that are beautiful to look at. And David has a real talent for taking collections of these molecules and putting them to scenes. I think he calls them molecular landscapes. And in this particular scene, it is bacteriophage T4 infecting an E. coli cell. and then replicating, taking over the cell. You can see that T4 degrades the host chromosome, and then you see lysis at the end. And I'm particularly attracted to it. It's beautiful. It's also depicting lysis, which is my favorite part of the bacteriophage replication cycle. I actually, these are shared in a collection under CCBY 4.0 license, so attribution. And so I printed this as a tablecloth that I use now for whenever we do outreach, so that you can just see in great detail all of these molecules,
nice.
And they're all colored, and I know what some of them are, and so we can point those out. It's really pretty. He has a lot of cool ones. He has one from 1999 that he did of a part of E. coli, and he updated that just a couple of years ago. So it's neat to see how the information that we've gained in that amount of time has allowed us to have more detail in these molecular landscapes. And there's all kinds of them. If you go to his Good Cell Gallery, you'll see many different kinds of molecules depicted in his watercolors.
I just love his work, and this is beautiful.
Yeah,
and this nice. And there's a story in this picture. I mean, he's got one cell that's lysing. He's got one cell that's in the middle of an infection with phage inside, but it's not lysed. And he has one cell that's just starting to get infected with the phage just attaching to it that came from the lysed cell. It's just very nicely composed.
It's nice, very beautiful. Yeah, I could see making a big something out of that. That's very cool. All right, we have a listener pick from Dirac who writes, you might appreciate this T-shirt. It says, the answer to every question will inevitably be, it depends. It's a shirt about powerlifting, maximum strength, squat, bench, deadlift. I don't know what that means. I know what a deadlift is, but I don't know what a squat
powerlifting
bench
is. Like Olympic weightlifting.
But what is a squat bench
as compared to bodybuilding, which is about building shape? And so squat, bench, and deadlift are three different.
Oh, they're three different things. Okay,
Three different things.
Got it.
No, nobody does a squat, bench, deadlift.
Nobody.
Not on purpose.
Oh, I see. I see.
Well, they might only do one of them.
Right.
And then
so it's a shirt about powerlifting from some of the leading practitioners. Athletics is a complex science and art, and where empiricism is entwined with anecdote, and the most resolute epistemology can distinguish itself only after decades and good luck, a difficult and complex practice entangled with the contingency of human ambition and mortality, and also Soviet sports supplements. Also, Dr. Griffin said Vincent should hit the gym. So, hey, here's his first step toward deadlifting for longevity and bone joint health, probably. Yeah, he did say that to me. I don't know. I don't remember why he said it the other day, but I'm trying actually to do some weight training. I'm doing deadlifts, squat, and bench press. Oh, the three things that are here.
There you go.
And I don't do it frequently enough because every time I do it, the next day I'm sore. And at some point, you're not supposed to be sore, right?
Well, no. Only for a while. Then you get used to it.
All right. That is TWiV 1067. Show notes, microbe.tv/TWiV. We'd love to get your support, microbe.tv/contribute. And if you have questions, comments, pics, please send them to TWiV at microbe.tv. Dixon Despommier, Racaniello.org, thelivingriver.org. Thank you, Dixon.
You're welcome. And thank you, Jolene, for the exploration of a world which I had never dreamed of before. Now I'll dream of it often.
Jolene Ramsey is at Texas A&M, College Station, Texas, and at ramseylab.vercel.app. Thank you, Jolene.
Yeah, thanks for having me. This is great.
I think you told a good narrative today.
Yep.
I really enjoyed
Nice
it.
story.
Wonderful
Yeah.
narrative.
Make a story out of it. It's great.
That's right.
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Alan Dove, alandove.com, turbidplaque.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me here at the Incubator in New York City. You can come visit if you'd like. You can just send an email, vincent@microbe.tv or at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. Thank you, Jolene. We appreciate it. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.